So today we are going to deal with pyruvate dehydrogenase complex. So it's also called PDH. Pyruvate dehydrogenase complex is a multi-enzyme complex which is made up of 3 enzymes and 5 coenzymes. So where this pyruvate dehydrogenase complex is functioning? This pyruvate dehydrogenase complex helps in the conversion of pyruvate to acetyl CoA. So pyruvate is converted to acetyl CoA. By this pyruvate dehydrogenase complex. So this pyruvate is formed from glycolysis in the cytosol, and this after the formation of pyruvate, the pyruvate is transported to the mitochondria from the cytosol. And this pyruvate is converted to acetyl CoA by a process known as oxidative decarboxylation. So oxidative decarboxylation causes the conversion of pyruvate to acetyl CoA. So this step, pyruvate to acetyl CoA, is a link between glycolysis and the citric acid cycle. So this is a link. This step is a link between the glycolysis and the TCA cycle or the citric acid cycle. The citric acid cycle is also called TCA cycle. So this pyruvate to acetyl CoA step, which is oxidative decarboxylation, is a link between glycolysis and citric acid cycle. The glycolysis happens in the cytosol of the cell, and the conversion of pyruvate to acetyl CoA takes place in the mitochondria. Now we can see what are the components of this multi-enzyme complex PDH. So you can see here. So there are 3 enzymes. So there are 3 enzymes. And 5 coenzymes in this PDH. So the 3 enzymes are pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. So the 3 enzymes of this PDH are pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. The 5 coenzymes present in the PDH are TPP, which is also vitamin B1, FAD, also called vitamin B2, NAD, also vitamin B3, coenzyme A, vitamin B5, and lipase acid. So these are the 5 coenzymes present in the pyruvate dehydrogenase complex. So now we can see the sequence of reactions of the PDH complex. So firstly, the pyruvate dehydrogenase enzyme, this has TPP as coenzyme. This enzyme decarboxylates pyruvate, that is, there is removal of carbon dioxide from pyruvate, and hydroxyethyl TPP is formed. Then the next enzyme, that is the second enzyme of the complex, dihydrolipoyl transacetylase, catalyzes the conversion of this hydroxyethyl TPP into acetyl lipoamide. So from this acetyl lipoamide, the acetyl group is transferred to coenzyme A to form acetyl CoA. Now this dihydrolipoyl dehydrogenase enzyme This enzyme, dihydrolipoid dehydrogenase enzyme, catalyzes the conversion of this lipomide, which is reduced, converts this reduced lipomide to oxidized lipomide by transferring the reducing equivalents from FADH2. So this reducing equivalents are transferred to lipomide reduced. So this H2 is transferred to FAD plus to form FADH2. So lipomide, which was reduced, is now oxidized lipomide. Then next, the FADH2, it transfers its reducing equivalents to NAD plus and forms NADH plus H plus. This NADH then enters the ETC or electron transport chain, or it is also called respiratory chain. So this NADH and H plus then enters the electron transport chain to generate the ATPs by oxidative phosphorylation. So these are the sequence of steps which is happening in the PDH complex, resulting in the formation of acetyl Co A. So the pyruvate, which is produced in the glycolysis in cytosol of the cell, is oxidatively decarboxylated to acetyl Co A. This acetyl Co A by oxidative decarboxylation. NAD plus is utilized here. NADH plus H plus is formed here. TPP is used. PDH complex is the enzyme which converts pyruvate to acetyl Co A. So this step, pyruvate to acetyl Co A, is a link between glycolysis and the TCA cycle or the citric acid cycle. So this is a link between glycolysis and TCA cycle. And the sequence of steps involved in the pyruvate acetyl Co A are this. Pyruvate is first decarboxylated, removal of carbon dioxide from pyruvate by the pyruvate dehydrogenase. So this TPP is converted to hydroxyethyl TPP. This hydroxyethyl TPP is again this dihydrolipoyl transacetylase convert this to acetyl lipomide. This acetyl lipomide gives out the acetyl group to coenzyme A to form the acetyl Co A. And this lipomide is reduced. This reduced lipomide gives away the hydrogen ions to FAD plus to form FADH2. And this is catalyzed by the enzyme dihydrolipoyl dehydrogenase. Now this reduced lipomide is oxidized to lipomide. Next, the H plus is transferred to NAD plus to form NADH plus H plus. And this NADH and H plus enters the respiratory chain to generate the ATP. So now we can see the regulation of PDH complex. How is this pyruvate dehydrogenase complex action regulated? So there are three methods by which this PDH complex is regulated. One is endpoint inhibition, which is also called feedback inhibition. Second one is phosphorylation, and third one is dephosphorylation. So this pyruvate dehydrogenase complex is active in the dephosphorylated state, and it is always inactive in the phosphorylated state. So the enzyme which causes the phosphorylation of the pyruvate dehydrogenase is protein kinase, that is PDH kinase. And the enzyme which always causes the dephosphorylation, that is removal of the phosphate group from the PDH, is phosphatase enzyme. So phosphatase is the enzyme which maintains the PDH complex in the active state. So this protein kinase, that is PDH kinase, it causes the phosphorylation of PDH. It causes the inactivation of PDH. So it is promoted by this enzyme, PDH kinase is promoted by ATP, NADH, Acetyl Co A. So this ATP, when it is present in large amount, it causes the activation of protein kinase, which causes the phosphorylation of PDH. That makes the PDH inactive. Then NADH and acetyl CoA also does the same thing. It causes the stimulation of protein kinase enzyme, which causes the inactivation. And this PDH kinase or protein kinase is inactivated by NAD plus when NAD plus is present in large amount, coenzyme A, and also pyruvate when they are present in large amounts, which leads to the inactivation of PDH kinase, which in turn causes the stoppage of phosphorylation. So this NAD plus, coenzyme A and pyruvate helps in the stimulation or promotion of formation of acetyl CoA. Acetyl CoA formation is promoted by NAD plus, coenzyme A and pyruvate. ATP, NADH and acetyl CoA causes the inhibition of acetyl CoA formation from pyruvate. Dephosphorylation is carried out by the enzyme phosphatase. This dephosphorylation, which is carried by the phosphatase, this phosphatase removes phosphate group. Phosphatase remove phosphate group by hydrolysis from the PDH complex. So thus the PDH becomes active. Then this pyruvate dehydrogenase complex is also inhibited by long chain fatty acids.